Thank you, Barbara, and thanks to whoever invited me. I'm glad I finally made it to Vienna, if only virtually. And thanks to Julia for helping to organize this meeting. I'm looking forward to meeting many of you over the next day or two. So I'm going to be talking about the evolution of transcriptome variation in organs of male reproduction in Drosophila. And what I'm doing is presenting three short stories. The first one's published. The second and third short stories are not yet published. In fact, analyses are ongoing, so some of the results are provisional, but hopefully I won't say anything that's dramatically wrong. You can look at the papers that eventually come out and then compare the seminar to the papers and see how wrong I was on certain things, I guess. All right, so let's just jump in here. So there are three major modes of transcriptome evolution listed here. Isoforms can evolve. We can see the evolution of transcript abundance, which is usually considered as a quantitative kind of endeavor. And then the evolution of novel genes also these novel genes make contributions to transcriptome diversity as well. I'm going to talk about mechanisms 2 and 3 here. And thinking about mechanism 2, typically, and I'm going to present some work along these lines, we quantify transcript abundance and try to say something about the evolution of transcript abundance from that quantitative data. In the first part of my talk, what I'm going to do is basically argue that it might be interesting to convert this quantitative data into categorical data or qualitative data. So you'll see if I can convince you that that's the case. And then in the second part of the talk, I'm going to be talking about a particular kind of novel gene, not gene duplications, which are extremely widespread, interesting, and important, but a relatively more rare, but to me, extremely interesting kind of novel gene, which we refer to as de novo genes. So that's what the second part of the talk is. Third part of the talk is going to be our initial foray into single cell data and how it informs us about transcriptome evolution. Okay, so I work on closely related species of Drosophila. In fact, the three species illustrated here in this small phylogeny. And there are lots of technical reasons for working on closely related fly species, which I won't go into. But there's also a very sort of important conceptual reason to work on closely related species, and in fact, to study polymorphism and divergence. And that is because we hope that by comparing polymorphic and fixed variation, we can eventually gain some insights into the mechanisms of evolution of whatever we're studying. I'll have to confess that for today's talk, I unfortunately can't say too much about population genetic mechanism. It really is mostly at this point an analysis of expression phenotypes, but we hope to eventually get there and use population genetics data to say something about mechanism. I'm going to be focusing on male reproductive tract tissues, although not quite exclusively. Here's a diagram of the dissected Drosophila male reproductive tract, melanogaster, in fact. And you can see the testis, which is obviously the sperm-producing organ. The other two organs I'm going to be focusing on are the accessory gland, a paired gland, and the ejaculatory duct, indicated on this slide. And their primary function is to produce seminal fluid proteins that are transferred to females during mating. When I use the phrase accessory gland during this talk, really I'm referring to the accessory gland plus the ejaculatory duct. So that's what accessory gland means when I use it today. There's a lot of reasons to focus on male reproductive tissues. There's a very large literature suggesting they evolve quite rapidly and often adaptively, and there's a lot of genetic novelties that seem to be expressed in the male reproductive tract. Not to say that there aren't other very interesting tissues, as interesting as the male reproductive tract, but this is a good place to go if you're interested in starting down this road of rapid adaptive evolution and genetic novelties. So that's what we've done. So the first short story I'm going to tell you came out in Genetics in September of last year, and it's a story about how old genes can evolve very new expression patterns. So here's this three species tree. And by the way, there are several things in this paper that I'm not going to mention, so I'm going to just focus on one small part of the paper. And that small part is gains of novel expression in particular organs of Drosophila melanogaster. So if we imagine we have this three species tree here, and we've got multiple genotypes of melanogaster sampled, we often sample at least one, often more than one outgroup strain for each of the outgroups. We estimate transcript abundance using RNA-seq in different tissues in this collection of genotypes. And then because these are very closely related species and evolution isn't extraordinarily rapid, we can use parsimony to reliably infer where the gene is expressed in the mel-sim common ancestor. And having done that through the use of the outgroups, we can then identify the gains of expression that have occurred in the lineage leading to melanogaster. Now, this table, which I took from the paper, is basically just a summary of the kind of data that we got. We have 6 melanogaster inbred lines, 3 F1s made from intercrossing those lines, and depending on the tissue, 1, 2, or 3 outgroup strains of simulans and Yakuba. Now, the tissues are accessory gland, AG, the testis, which you know. SG stands for salivary gland of the third larval instar. So the reason we picked that is because we thought it might be interesting to start understanding if there are unusual properties of the accessory gland in terms of its evolution. Are those properties a function of its role in male reproduction, or is it a general property of glands, right? So since it's a gland and it's reproductive, you can't tell if there's something unusual about it, whether it's a gland phenomenon or a male reproductive tract phenomenon. So we hope to gain some insight by including that tissue. Heads are female heads, and we picked those simply because they're easy to collect, and there's a vast literature of transcriptome studies on female heads. And the first instar larva we picked because it's very well defined developmentally, it's easy to collect, and there just wasn't much data of transcriptome evolution on this developmental stage. So those are the tissues and the genotypes that we used. And let me explain to you what we were looking for here. So this is a little diagram which is showing us in the first vertical blue column a polymorphic neomorph. I'm going to refer to these gain of expression variants as neomorphs, even though we don't have data showing that they're gain of function variants. So the first blue column is a polymorphic neomorph, and the second one is a fixed neomorph. So how do we categorize genes into these two categories? Well, if we look at the first vertical blue column, we see that there is a gene that's had its expression estimated in a tissue in 2 Yakuba strains, 2 simulan strains, and 2 melanogaster strains. The open star indicates that there's no evidence of expression, right, in the strain. So what we have is neither Yakuba strain expresses the gene in the tissue we're thinking about, neither simulan strain does, and in the first column, one of the 2 melanogaster strains expresses the gene. So we would identify this as a polymorphic neomorph, and naturally, if both melanogaster strains express the gene, we would categorize it as a fixed neomorph using parsimony. So what we've done here is we've taken, as I mentioned earlier, quantitative data on TPM's expression estimates, and we've converted it to categorical data. So how do we do this? Well, we declare a gene to be not expressed in a line if its TPM is less than 0.1. So this is a very low level of expression. In the literature, it seems to be that it's very conservative, so I think that was a reasonable thing to do. And then to call a gene as expressed, we call it expressed if it's greater than TPM greater than 2. This also seems quite conservative in many papers, including some from our lab. We call genes expressed with TPMs of 1. So we think this is a conservative way of categorizing genes as not expressed or expressed in a particular tissue and strain. And we have investigated altering these cutoffs to see how they affect the results, and they basically affect them as you would expect from a quantitative point of view, but the overall patterns seem to be pretty robust to the arbitrary cutoffs that one might choose. All right, so that's what we've done using the data set that I just described. So what did we find? So we identified 79 polymorphic neomorphs and 6 fixed ones. So a total of 85. Not a lot. We looked at several thousand one-to-one-to-one orthologs in these 3 species. Most of these polymorphic neomorphs were expressed in only 1 line, but that still leaves a lot that were expressed in multiple melanogaster strains. So it's not strictly a phenomenon that one can write off simply as a sort of deleterious variation that's appearing at low frequency in these melanogaster populations. And as you might expect, based on the literature, the accessory gland and the testis express the greatest number of neomorphs. So that's consistent, like I said, with a lot of literature about novelty in tissues of male reproduction. So I want to show you a little bit what these neomorphs look like in terms of the actual data. Oops. Okay, so here is a table from our paper displaying the TPMs of the 6 fixed neomorphs. So the melanogaster TPMs are listed under the demelanogaster column. The F1 TPMs are listed next to it, and then you can see the outgroup TPM. So this is very, very low level of outgroup expression, close to zero or actually no detectable expression in many cases. And you can see that these various genes are expressed quite abundantly in at least some cases. And robustly in all cases, and that the TPMs of the inbred lines are recapitulated for the most part in the F1. So this, we think, is a fairly robust phenomenon that we've documented here. You can see that 3 of these are expressed in the testis and 2 in the accessory gland. For a lot of these genes, there's not a lot of known functions that would give you an obvious clue as to what they're doing in the tissues in which they've acquired novel expression patterns. But there's a lot of interesting evolutionary questions and functional questions now that we have this gene list. A good example to think about is LMD, the gene at the bottom of the first column. It's called lame duck, and it's a pretty well-studied transcription factor that's expressed in several tissues. The kinds of questions we're interested in answering are, when this gene is expressed in the testis, is it recruiting novel components into downstream pathways, or is it just simply recruiting its ancestral genetic network into the testis? How is this all working? Are there any phenotypes associated with this expression at all? These are really important problems that we hope illuminate the evolutionary phenomenon that we've documented. And we're engaging in some RNAi experiments with these genes to try to figure out what's going on. I should add, with some help from the Vienna Drosophila Stock Resource Center. All right, so those were the 6 fixed neomorphs. I don't have a lot to say about the mechanisms. We'd love to understand the role of selection in generating these phenotypes, but we really don't have a lot of insight into that yet. Here are some of the polymorphic neomorphs that are expressed in the testis. So here we have 3 genes where the TPMs for the individual melanogaster inbred lines are displayed, as well as the outgroup TPMs, which are all 0. You can see that these are intermediate or high frequency phenotypes where, again, there's fairly robust expression in some lines in the testis and really no evidence of expression in other lines. So these are extremely young phenotypes that are still spreading or have spread through Drosophila melanogaster populations. Okay, now it's really interesting to think about how these evolve in terms of functional and evolutionary questions. But I think one key component of trying to understand what's going on here is to try to say something about the genetic basis of these expression phenotypes. So we took a first crack at trying to do that by doing a small-scale allelic imbalance experiment, which I'm going to show you data for in a moment. These experiments, basically, you take TPM estimates or gene expression estimates from the parental strains, and then you cross the parental strains and estimate again gene expression in the F1s. And you also, in the F1s, assign the reads that you collect from the F1 tissue as to having been derived from one parent or the other. So this is a pretty widespread technique in the genetics and genomics literature, and I'm not going to go into great detail, although there are some technical challenges to getting sort of reliable estimates in these experiments we've learned. What we've discovered in these experiments is that roughly 30 to 40% of the genes show complex underlying genetics. So we don't know how complex they are, but they're not extremely simple. So there are cis and trans effects. The number of trans-acting factors affecting expression is a complete mystery at this point. Could be many, it could be only one, we don't know. But for a significant percentage of genes and crosses, we observe pure cis effects. So 14% of the polymorphic accessory gland neomorphs and about a fourth of the polymorphic testis neomorphs can be explained simply by cis-acting variation that's leading to expression differences among genotypes of Drosophila melanogaster. And what I want to do now is show you data from one of these really interesting polymorphic neomorphs, phyllopod. So these 3 columns of data represent data from A and D is one pair of inbred lines, B and E is a second pair, and C and F is a third pair of inbred lines. So we'll just focus on A and D because basically the conclusions are exactly the same for the other two experiments. You can see in panel A that we have a TPM estimate for RAL34 and RAL307. RAL304 expresses the line robustly. RAL307 shows no evidence of expression of this gene in the testis. The bar in the middle is the TPM estimate for the F1, which is exactly intermediate between the two parents, consistent with additive effects for this expression phenotype. Now, if we look at panel D for this pair of lines, what we've done is taken those reads from the F1, the intermediate genotype in panel A, and we've basically used SNPs to assign the reads to either the RAL304 parent or the RAL307 parent. And what we discovered in this case is that essentially all of the reads in the F1 derive from one of the parental chromosomes only, the chromosome that expressed the gene robustly as a homozygote, RAL304. This is complete allelic imbalance, so the only real interpretation of this is that there's some cis-acting variant that's probably tightly linked, although it could be far away, it could be, but it's probably tightly linked to the gene phyllopod, and it's driving expression in the testis of Drosophila melanogaster. The other genotypes gave us exactly the same information. So this is a really cool case where we have a gene that seems to have had that near it was born a cis-acting regulatory element that is driving robust expression in the testis, and that variant or variants, they have risen to intermediate frequency in melanogaster populations. Now, this is the kind of situation where you could hope to try to use population genetics data to try to say something about the role of directional selection in the spread of this gene, but we really haven't got any traction on that question yet. And the functional consequences of this expression in the testis are very interesting to think about, but we don't have any information about that yet either. So we're basically at the situation where we have documented a lot of really interesting novel phenotypes, and we don't yet have functional or really any useful population genetic insights into how these phenotypes originate and spread. So obviously those are our goals for the next year or two, to try to really gain some more insight into these questions in Drosophila melanogaster. Now, I think there's a lot of interesting questions, but this kind of experiment and a lot of the work that we've been doing lately has just made me sort of ponder about a species, melanogaster, which we think has its history is dominated by selection, both genomically and phenotypically. There may be a little bit of room for drift in this species, but selection is clearly very important. And it just raises this very interesting and general question, I think. Is every aspect of gene expression in an animal like this, where its genome seems to be dominated by selection, is every aspect shaped directly or indirectly by this selection? Is there a role for drift, for example, or deleterious alleles generating all of these novel phenotypes? I mean, this is something that I think is very interesting to ask, not only in melanogaster, but more generally. Okay, so that's short story number one. That was primarily the work of Dr. Julie Cridland, who's a project scientist in my lab. And she's also primarily responsible for this short story, which is unpublished and analysis ongoing, which is to ask about novel genes in the accessory gland of Drosophila melanogaster. Okay, so let me briefly tell you what de novo genes are. They're not new genes that have originated by duplication, which is how most new genes originate. They're an unusual class of genes that we think come into existence from ancestrally non-coding sequence. A plausible model for their origin is that spurious transcription occurs throughout the genome occasionally, tends to be suppressed by natural selection because it's, we would think, deleterious, but occasionally some of this spurious transcription has the proper signals for processing and actually ends up conferring a fitness benefit on the individual that carries whatever variant or variants lead to this expression. And these novel genes can spread through populations. Okay, so what would a de novo gene look like phylogenetically? Well, here's an imaginary phylogeny, not imaginary, for real species, but depiction of a tree and what a de novo gene would look like. So we imagine that we've identified, based on annotations, what we think is intergenic sequence, for example, in all 4 of these species. And yet, you know, the annotations are incomplete, and when we look at transcriptomes from, let's say, Drosophila yacuba, we see evidence in this supposed intergenic sequence. We see evidence, good evidence, in fact, of a transcript and a gene. But no matter how hard we look in transcriptome data or do sequence analysis in the outgroups, we can't find any evidence that there's a gene in the homologous location of these three other species. Okay, so there's a couple of explanations for this phenomenon. One is that ancestrally this is a piece of intergenic DNA, and it has sort of acquired the status of a gene in Drosophila yacuba through the mechanism I just described, perhaps. Another possibility is that this gene was present in the ancestor but was lost 3 times. Lost in melanogaster, lost in erecta, lost in Ananassi. That's consistent with the data as well. And a third possibility, of course, is that we haven't worked hard enough to find the expression of this gene in these 3 other species other than yacuba, and it's expressed at a very low level or in very few cell types or in an environment that we haven't assayed in the laboratory, for example. And the gene really is there, but we haven't discovered it. So this is a formal possibility as well. I mean, what we've done in our work is try to be as conservative as possible, use as much data as we can, use conservative cutoffs in every case to try to reduce the likelihood that what we have here is an artifact of extraordinarily low expression in the outgroup, in the non-focal species, I should say. We've also tried to use all kinds of cutoffs to sort of make sure we look at robust transcription in these focal genes that we suspect are de novo genes. For any individual gene, we can't rule out that it wasn't lost 3 times, for example. So, I mean, overall, for any individual de novo gene or putative de novo gene, you cannot rule out some alternative hypothesis. But the fact that we've done this, I think, pretty carefully, and the fact that we've discovered a fairly widespread phenomenon suggests that it's not all artifactual, that these are real entities, and there's evidence, obviously, from other laboratories that have been studying this phenomenon that it's occurring in other lineages. As you would expect, there's nothing particularly special about the biology of Drosophila in these terms. Okay, so that's what a de novo gene is. So let me tell you about some of our early work. The early work was strictly comparative, so no within species data, and focused on the testis and the accessory gland. That's where we discovered this phenomenon in the first place. More recently, in 2014, we published a paper focusing on the population genomics of de novo genes in melanogaster, focusing on the testis. And in that experiment, we looked at 6 inbred lines of melanogaster, in fact, the same 6 that I discussed in the first part of the talk, and we observed a lot of de novo genes, roughly 250. Over 100 of them were fixed between species, so none of them were expressed in simulans, and all the melanogaster strains expressed these genes. And a lot were segregating, so some strains expressed a gene and some strains did not. So the general properties of these genes are they're short and simple. The rare genes, as the genes expressed in only one strain, tended to be shorter and expressed at lower levels compared to genes expressed in multiple strains. They're strongly underrepresented on the X. That's nice because it's consistent with the literature of old or ancestral genes in this species. We suspect that they code for small proteins, but we don't have direct experimental evidence of that yet, and that's an important part of this whole field, to try to understand the protein coding capacity of these genes. For the polymorphic genes, we found that cis effects were the major cause of the expression variation. So the genetics is pretty simple for these testis-expressed genetic novelties. And we did do some population genetic work in that paper to suggest that there's some evidence for directional selection in the spread of these genes through melanogaster populations. Now, obviously, there's a lot of extremely important functional questions, some of which we're working on right now, to try to illuminate the evolutionary explanation for these genetic novelties. That's a very important part of all de novo gene work at this point. But there are some very general evolutionary population level questions too. Is the testis highly unusual? And in general, de novo genes expressed in different tissues, do they have different attributes functionally, in terms of their structure and organization, in terms of the evolutionary processes that explain their existence. So these are very general questions. And we've taken a stab at starting down this road by looking at the accessory gland, which I mentioned earlier in the talk. It produces seminal fluid proteins that are transferred to females. It's a pretty simple tissue compared to the testis, but it still has very interesting biology and lots of evidence of rapid and adaptive evolution. So it's simple, but still very interesting. And it's also interesting to think about comparing the evolutionary dynamics of the accessory gland and the testis, because there is this question about how unusual is Drosophila male reproduction in terms of rapid adaptive evolution and evolutionary novelties and so on. One nice thing about comparing the accessory gland and the testis is that they both function strictly in male reproduction, and yet one is somatic and one is primarily germline. And so by comparing these, maybe we'll start to get some insights into the relative importance of selection on somatic versus germline functions in terms of the evolution of novelties. Okay, so the data set that we use for this is very similar to the one that I just described to you, so I'm not going to go into it, but let me tell you a little bit about what we did to find these de novo genes. So what Julie did is she took a whole bunch of reads from the accessory gland and ejaculatory duct of the melanogaster inbred lines and did a large-scale de novo transcriptome assembly from these reads and came up with, as you would expect, a giant pile of transcripts. And what we are interested in are melanogaster transcripts that aren't associated with any melanogaster annotations or annotations in other species, and in fact for which there's no evidence that they're expressed in the other species. So we take these de novo assembled transcripts and then we just screen out everything that matches an annotation in any of the three species. Or if it's unannotated in melanogaster, we often were able to recover a transcript in the outgroup. So these are just ancestral unannotated genes, which could be interesting, but we threw those out too. Then for the remaining transcripts, which we have evidence for expression in melanogaster, they're not known genes, and there's no evidence of expression in the outgroups from any data that we have. We filter them by proximity to known genic elements. And the reason we do this is because we want to reduce the probability that we're looking at unannotated exons of known genes or looking at something like read-through transcription, for example. So we're trying to reduce those kinds of artifactual findings. When we filtered out all of that, we take what's left, we go back to the inbred lines and then estimate transcript levels. And we keep those genes where TPMs are at least one in at least one melanogaster strain. So that's kind of the pipeline for identifying these candidate or putative de novo genes, in this case expressed in the accessory gland. All right, so we did this, and what did we discover from looking at all this accessory gland data? So we discovered a very large number of novel transcripts in the accessory gland, 247 roughly. This is the part of the analysis that's ongoing, so these numbers are probably going to change a little bit between now and when the paper comes out. But 247 genes producing about 296 transcripts. From that, you can divine that most of the genes have only one transcript, and in fact, most of the transcripts are just one exon. So these are short, simple genes, consistent with what we've observed before in the testis and what other people have observed in de novo gene literature. It's still kind of interesting, though, to see that several of these very, very young genes have multiple transcripts and sometimes 2, 3, or 4 exons. So even though they're very young, they're not all extraordinarily simple. Okay, so in this analysis, we actually looked for genes in introns, obviously large introns based on our filtering criteria in terms of proximity to annotated genic elements like exons. We didn't do that for the testis. I mean, you might actually expect more de novo genes to be born in introns because there's just more transcriptional activity in introns. The reason we didn't do that in our earlier work was that we were seeking to be very, very conservative. And there are some technical challenges working with intronic data. But here we decided to take a look and see what we can see. So 176 of these genes were intronic and 71 were intergenic. That 71 intergenic genes is directly comparable to what I told you for the testis, because it's the same strains and it's the same genomic space that we use to look for these genes roughly. So there are many more of these de novo genes expressed in the testis than in the accessory gland. On the other hand, the transcriptome diversity of the testis is much greater than that of the accessory gland. And so we're still trying to do some analysis to sort of characterize the contribution of these novel transcripts to transcriptome diversity. I think it's safe to say there are more of these in the testis, expressed in the testis than the accessory gland, but that they actually still make a pretty significant contribution to the accessory gland transcriptome. All right, so you can divide up these genes into those expressed in only one strain, two strains, three strains, and so on. And for the strains expressing a gene, which requires a TPM of at least one, you can then calculate the mean TPMs. And that's what's displayed here. And what you can see is that genes that are expressed in only one strain are expressed at a lower level than genes expressed in multiple strains. That's also very consistent with what we observed in the testis. So we think at this point that seems pretty robust. It's probably a general phenomenon. Genes expressed in only one strain tend to be expressed at lower levels than genes expressed in multiple strains. I think there's some interesting possible evolutionary interpretations of that, which I am not going to go into right now. But that's the data. Now, what about the lengths of these? Well, these genes tend to be short. I told you they're mostly single exon genes. There are some pretty big ones in there, though, and we're going to take a look at some of those a little bit more carefully. The median is a little bit under 500 base pairs. So these are very much shorter than the transcripts that we identified in the testis that we think evolved de novo, maybe about half as long. So that seems to be an interesting difference between these two tissues. That's a bit of a provisional conclusion. I mean, things are going to change a little bit, but I think this is going to be a sound conclusion at the end of the day. Now, one of the things I mentioned in the previous slide is that singleton genes expressed in only one melanogaster genotype tend to be expressed at lower levels. They also tend to be shorter, and that too was observed in the testis. So generally, there are lots of similarities between the testis and the accessory gland. But there are some differences. But what I want to show you next is what I think is the most obvious and biggest difference between these two tissues in terms of the attributes of these de novo genes, and that's the frequency distribution. If you look at the frequency distribution, and I'm not using that in the population genetic sense, I'm using it as a description of the number of inbred lines that express these novel genes. There's many of these de novo genes expressed in only one strain in the accessory gland, and very few, only 4 out of 247 were expressed in all 6 melanogaster strains. That's very different from the testis, where there are relatively fewer expressed in only one strain and many more that were fixed. Now, one of our main tasks going forward is going to be to discriminate among the different possible explanations for this difference between the tissues. Is it that the accessory gland is more tolerant, for example, of deleterious expression? Is it the case that some kind of interesting natural selection is driving these novel genes in the accessory gland into populations, but the selective environment changes so quickly that they rarely fix? Is it the case that a lot of the expression in the testis has no fitness effects, for example? Sorry, in the accessory gland has no fitness effects. You know, these are very interesting evolutionary questions to which we really don't have any answers yet, but this is one of the main goals we've got, and I think the whole field has to grapple with the population genetic mechanisms and the functions of these de novo genes, which we're all sort of struggling to do, but on the way down that road, I think. Okay, so there's the major difference between these two tissues. Now, when you look at the novel transcripts expressed in the accessory gland, it's very apparent that they're non-randomly distributed on a chromosome-arm scale in 2 ways. First, the X chromosome is underrepresented, so that's compared to the autosomes. So that's actually consistent with what we observed in the testis and consistent with patterns of ancestral male-biased genes that tend to be underrepresented on the X. So that's a nice consistency. I think the other thing you can see is if you look at the autosomes, 2L, 2R, 3L, and 3R, you can see that we haven't done the formal analysis yet, but these are very likely going to be heterogeneously distributed. So some arms have significantly more than others, more so than you would expect. There's some, I don't want to say random distribution, but distribution based on the available sequence space in which de novo genes could evolve on each chromosome arm. So that's different from the testis, which did not show such a pattern. And it's very interesting to think about why this might be. Maybe the domains, excuse me, maybe the domains of expression of the accessory gland tend to be clustered along chromosome arms more so than for the testis, and maybe that's where these de novo genes are originating. So that's an interesting possibility, which we think we can get at. I also want to mention that when you look at the transcripts and where they're distributed in the genome within chromosome arms, you see that there is some evidence of small-scale clustering, which is consistent with what I've just described. There are a few cases where there are a couple of these novel transcripts that are very close together in the genome sequence. So that's interesting. We have no statistical results about how unexpected that observation is, but my sense, given the relatively small number of these transcripts, only 247 genes, is that that's likely to be significant when we finish the analysis. So that's different from the testis, and understanding that difference is going to be interesting and important. Okay, so one of the big questions in this field is, are de novo genes protein coding or not? And it's actually not a question that's answered yet. We haven't answered it yet, and I don't think it's been answered very well in general. So what we decided to do excuse me, is use a program called CPAT, Coding Potential Assessment Tool, to take these transcripts and see what it tells us about the probability that they're coding or non-coding. Now, this is a software package that was designed exactly to do this, to categorize transcripts as coding or non-coding, and it's been trained on melanogaster annotations. So it's, and some other species too. But we thought it would be kind of interesting to try it and see what we found. On the x-axis of these two figures, you see the coding potential score, basically, that CPAT returns. And if it's less than 0.39, it means that it's very likely to be a non-coding gene that's producing this transcript. So you can see the distributions of the scores for the intronic and intergenic sequences are similar, but more importantly, almost none of them are predicted to be coding. So at least according to this tool, relatively few or almost none of these actually are protein coding genes. They're mostly non-coding genes. So if that's true, it's interesting because the accessory gland produces seminal fluid proteins primarily. It's a big part of its function, which are secreted, obviously. They're proteins that are transferred to females during mating. And yet, if this is correct, the novelties that are originating are primarily non-coding. So, that raises some pretty interesting questions as well. But we were concerned about this conclusion from CPAT because these genes tend to be very small. And so we were worried about whether the algorithms used by this program were basically incorporating the lengths of the transcripts in a way that made the program more likely to make errors. So what we did is we took all the annotated genes, both coding and non-coding, and we put them through this program and generated the CPAT scores. And here they are in these two figures for coding on the left and non-coding genes on the right. And these bins represent 100 base pair gene size increments. So the first bin is genes that are 100 to 200 base pairs long, then 200 to 300, and so on in 100 base pair increments up to 2 kb. What you can see is that for the annotated non-coding genes, so let's just assume for the moment that the annotations are correct. For the annotated non-coding genes, the CPAT scores are very low. In the vast majority of cases, well below 0.39. So gene size doesn't seem to be having a very big effect. It maybe has a small effect, but not a very big effect on the probability that the program will, if you think the annotation is correct, mischaracterize a coding gene as non-coding. Now on the left, we see all the annotated coding genes, and here you see a different story, which is that, especially at the tail of the distribution of gene size for the really small genes, CPAT, if you think the annotation is correct, CPAT is making a lot of errors. It's calling small annotated coding genes to be non-coding. So this made us a little worried that perhaps we're not getting the right answer from this piece of software. And we still obviously don't know. It takes real experiments to figure out what's going on here. But one thing I'll point out is that if you look at that left tail of the distribution, let's say transcripts that are 300 to 400 base pairs that are coding, or 400 to 500 base pairs. I mean, that's kind of in the area where we are in a lot of our de novo accessory gland express transcripts. And CPAT makes errors, but its median score is really quite high. So basically, we're in a bit of a quandary here where we don't, this didn't really help us very much, I think, in the end, understand what's going on. And like I said, real experiments need to be done to figure this out. You can't use the presence of an open reading frame really as a way of determining unambiguously whether a transcript is coding or non-coding, because the vast majority of non-coding genes carry small orphs. So this is still up in the air, but one thing I will point out is that if we take the best orphs from our sequence analysis, which actually CPAT produces using getOrph, and we put these orphs through a program to predict the existence of signal sequences, we find none or almost none of these accessory gland expressed transcripts have signal sequences. What does that mean? It means they're very likely not secreted proteins. And again, we get back to this point that all of these novelties expressed in the accessory gland, they clearly, I don't want to say clearly, it seems very unlikely that they're new seminal fluid proteins that are secreted and transferred to females. So what are they doing? I mean, this is an extremely interesting question, again, that we hope to get at through some genetic analysis in the future. But the novelty in the accessory gland does not seem to be mostly novel seminal fluid proteins. So I guess one main conclusion I wanted to point out is that this polymorphism of these novelties in the accessory gland is way greater than the polymorphism in the testis as a proportion of all the novelties that we documented. And that's true of the neomorphs, if you remember. I didn't actually mention this while I was talking about it in the first part of the talk, but a greater proportion of the neomorphs expressed in the accessory gland are polymorphic as compared to fixed relative to the testis. And in fact, we've documented some very interesting gene presence-absence polymorphism for accessory gland bias genes. And so it does seem quite general at this point, I think, that the accessory gland has way more polymorphism for these very unusual this unusual genetic and phenotypic variation compared to the testis. And that is something that we need to address. So we hope to address some of these interesting questions by getting at the genetic basis of these novel expression phenotypes and really trying to get a population genetics handle on it. We have to do the first, get at the expression genetics, I think, to get at the population genetics. And we have to do some functional experiments, and the tools in melanogaster make it fairly straightforward to do, and we're working on some of those right now. We desperately hope, that the results from these functional experiments will help us understand what we're seeing from an evolutionary point of view. That's really the point of doing these functional experiments. So we hope that that works out for us. All right, so the third part of my talk is the work of Alelic, who's a graduate student in my lab, doing really terrific work, primarily on the accessory gland in Drosophila. And he decided to undertake a single cell analysis of the accessory gland. Now, the fly literature for many years has been rich with whole animal transcriptome data. It's actually not particularly great data. I mean, if you were going to try to understand transcriptome evolution, basically grinding up a whole animal and sequencing it is kind of one of the sort of, it's a very blunt instrument, let's put it that way. But we and other people, many other people, have been able to discover some interesting things from that kind of data, even though we all knew, I think, that we should have been working on individual tissues from the beginning. The same kind of argument that, variation among organs is obscured when you grind up a whole animal and sequence its transcriptome. That very same argument applies to the organs themselves, because most organs are complicated mixtures of tissues and cell types. And so it's inevitable that when you grind up an organ and sequence its transcriptome, there's a lot of underlying heterogeneity, both in transcriptome variation, possibly evolutionary mechanism that's hidden from you when you do those bulk organ experiments. And so, I think this is one of those cases where the technology of single-cell sequencing has advanced to the point where now we're really finally, after many, many years of transcriptome work in Drosophila from an evolutionary point of view, finally getting to the level that's the correct, if you want to think about it, the correct level for understanding expression evolution and its underlying population genetic causes. So thankfully, we're finally at the point after many years where I think we're going to really get the data that we need. Whether we get the good answers to our questions remains to be seen, but we're going to get the right kind of data at least. So I want to remind you, tell you something about the accessory gland that I didn't mention earlier, which is that it's a simple gland, which I did mention. It's composed primarily of two cell types, the main cells, which is the vast majority of cell types, and the secondary cells, which are found at the tips of the glands. Very, very rare compared to the main cells. They're, excuse me, morphologically distinct. You can see this image on the right hand here is made by an undergrad in my lab, Yoko Takashima, who's working on accessory glands. And you can see those large dark circles. Those are large vacuoles associated with secondary cells. So you can see they're quite easy to distinguish from main cells. And here they are at the tip of the gland. Now, besides being morphologically distinct, there's some transcriptome differences between these cell types that we know about, but mostly they're uncharacterized. There's some genetic data, though, that suggests that even though they share production of certain transcripts, they also have some distinct functions. And the way that this is described primarily in the literature is that the main cells initiate the female post-mating response, ovulation, position, proper storage of sperm and sperm storage organs, and so on. Whereas the secondary cell products that are transferred to females are thought to play a more important role in maintaining the long-term post-mating response. So, but not a lot's really known about, in detail, about the different cell types. And so we thought it would be interesting to actually do some single cell work to not only really characterize these cells for the very first time, and I should say that we included the ejaculatory duct. I forgot to mention that. Not a lot's known about that, so we decided to include that tissue as well. We decided to see what we could learn about the biology of these organs from the single cell analysis, and then more perhaps relevant to the lab's main line of work, to sort of investigate patterns of transcriptome evolution across cell types and lineages to try to see if we could see evidence of heterogeneity of processes of different cell types, and then to understand how the biology of these cell types intersects with the sort of prevalence of protein adaptation of genes that show biased expression. So basically, is there evidence that adaptive protein evolution is more important for some cell types than for others? So that's kind of where we're trying to go here. So, excuse me, what Alex did is he collected accessory glands and ejaculatory ducts of all three species, pulled them, isolated nuclei, and then these nuclei were sequenced on the 10x Chromium platform. Then the reads that we obtained from these nuclei were used to, based on SNPs, to assign these nuclei to different species. So it was fairly obvious which nuclei came from melanogaster, which came from simulans, and which came from Yacuba. So after that was done, we took the reads from the nuclei from each species and then subjected them to a fairly common bioinformatics pipeline for dealing with single cell data. So that's Alex's work, and he's done it very carefully and very well. So let me show you some of the data that we generated from this. So on the left, you see a UMAP plot, which is a dimension reduction visualization technique for seeing basically how many different cell types you have in your single cell data. You can see right here in the middle of the UMAP plot is the large cluster. That's the main cells. And we identified these cell types by what's known in the existing literature about genes that are expressed in a very biased way in these different cell types. You can see in the upper right the secondary cells, relatively few, and you can see in the lower left the ejaculatory duct cells. So this is telling us a couple of things. First, there doesn't seem to be any clustering or different types of ejaculatory duct cells, at least in this data set. They're all in one cluster. And then in the right side of the figure, you can see the numbers of different cells obtained from Drosophila melanogaster. And these numbers, especially main cell versus secondary cells, are basically spot on what you would expect from the literature, suggesting that this whole process didn't result in a very biased sampling of nuclei from these tissues. Okay, so that's good. There are other attributes of the data that I'm not showing you that suggest that we have a very, very sort of typical good quality data set here that we can use for understanding transcriptome evolution. All right, so let me tell you about a few of the findings that Alex has made so far. So you can characterize a marker gene as being basically a cell type biased gene. And a lot of the SFPs, right, are the seminal fluid proteins, are a lot of the proteins that people focus on when thinking about the gland itself and its role in Drosophila biology. But there are SFP markers, so SFPs that show biased expression to different cell types, and there are non-SFP markers, so genes that are not seminal fluid proteins but that show biased expression. What this bit of data is showing is that the main cells seem to be quite specialized for SFP production. So a lot of the markers that characterize main cells that show main cell biased expression are SFPs, much more so than for the other two cell types, the secondary cells and the ejaculatory duct cells. I think this is pretty much what people expected in the literature, but because this is the first single cell data set from the accessory gland, it hadn't been conclusively demonstrated. And this, in fact, I think supports that notion that the main cell seems to be specialized for the production of seminal fluid proteins, although there are SFPs, obviously, that are biased towards these other cell types, too. And then there's a lot of non-SFP markers, and that's interesting, too, to think about, well, what are these biased genes doing to sort of shape the specific functional properties of these different cell types? That's an interesting question. Now, one of the things you can do with these data is identify novel marker genes. So these are genes that show biased expression towards particular cell types, which, at least in many cases, may confer, as I mentioned, specific biological properties to these cells. So for secondary cells, there were a few known markers, and Alex has discovered a few more. So this is just very sort of important for people that are interested in understanding the functional aspects of secondary cells, right? You have to know a lot about the biased genes expressed primarily in secondary cells to give you some clues about what they're doing. So this is just discovery-based science of new genes that are biased to expression in secondary cells. And also he discovered a bunch that were previously unknown that show biased expression in the ejaculatory duct cells. So this is just, as I mentioned, just cool novel biology associated with these different cell types. All right, let's get to the evolutionary stuff for the last few minutes. So here I'm showing percent genes differentially expressed in 3 different cell types for 3 different species comparisons. Mel versus Sim comparisons for 3 cell types on the left, in the middle, Mel versus Yakuba for 3 different cell types, and Sim versus Yak. So the Mel versus Yak comparison and the Sim versus Yak comparison is a greater time scale than Mel versus Sim, but the time scale is the same for Mel, Yak, and Sim Yak. So there's the same time scales. They have the same common ancestor. All right, so that explains why there seems to be more transcriptome divergence for the Mel Yak versus the Sim Yak comparison. There are more divergent species comparisons than Mel versus Sim. That makes sense. But the thing that first jumps out at you, and is the most obvious thing here in the data, is that the ejaculatory duct cells, transcriptomes, are evolving very much faster than the transcriptomes from the other 2 cell types. Why is that is a really fascinating question. It's a previously unknown phenomenon. So now we can start thinking about why this might be the case and maybe trying to get some answers from population genetic approaches to this question. The other thing I want to point out here is that, again, the Mel-Yak and Sim-Yak comparisons are the same timescale, but the ejaculatory duct divergence is quite a bit greater for the Mel-Yak comparison than the Sim-Yak comparison, consistent with accelerated transcriptome divergence in the melanogaster lineage specifically. So in our paper that we hope to publish in a relatively timely manner, I'll have more quantitative statistical analyses to present to you about these patterns, but I think they're fairly robust. Now, I mentioned that I wanted to intersect adaptive protein evolution data with these cell-biased expression patterns. So we're doing that not by using individual statistical inferences about, yes, a gene is evolving adaptively at the protein level, it has a p-value less than some value. But we're going to use alpha, which comes out of the MK sort of world, to use it as a sort of global estimator of positive selection, so directional selection. So alpha is the proportion of divergence resulting from selection for proteins, and if it's positive, that is, if alpha is greater than 0, it's consistent with directional selection. If it's less than 0, it's consistent with a couple of models, perhaps mutation selection balance. All right, so the first numbers here represent the median positive alphas for all cell-biased markers. And you can see that they're all pretty large, not unexpected given the literature on Drosophila, but that the secondary cells seem to be undergoing a lot less adaptive protein evolution than the other two cell types. So again, that's a novel finding. Of course, you couldn't really do that without this kind of single cell data. And there's about as much adaptive protein evolution in the ejaculatory ducts as in the main cells. So that's interesting as well. Now, this could be driven by variation in SFIP production because seminal fluid proteins are known to be evolving very rapidly and in many cases adaptively. But when you do this analysis for the non-SFIP markers, the cell-biased genes that are not SFIPs, you basically get the same result. So this seems like a pretty general finding that for some reason, secondary cell-biased genes seem to be experiencing less adaptive protein evolution compared to the other two cell types. And this can't be explained solely from variation in selection on SFIPs. Okay, so that's an interesting novel finding too that we hope to investigate a little bit further. I guess the final thing I wanted to mention is that there's a very interesting question in the literature about the correlation between differential gene expression, that is divergence between species in gene expression, transcript abundance, and adaptive protein evolution. How connected are these things? There's actually not a lot of really good literature on this, but it's a really important question. This question about this differential gene expression, how much it's driven by selection, is also very important, and that's not really addressed very well in the literature as well, and I'm not going to do so here, but we do have differentially expressed genes from the data I just showed you. And what we've done is basically to combine a bunch of MK data with these differentially expressed genes, and we find that differentially expressed genes do seem to have a greater proportion of their amino acid differences between species resulting from directional selection. So we think that's a pretty interesting, somewhat new finding that we hope to explore a little bit more too as we delve into the data a little bit more. Finally, putting the two parts of the talk together, parts 2 and 3, you might be wondering, what about those accessory gland de novo genes you described? Where are they expressed in the accessory gland? Well, they seem to be primarily expressed in the main cells. They're significantly main cell biased. And I find this really interesting because we really don't think they're seminal fluid proteins. They're novel genes that are not seminal fluid proteins, probably. The main cells are specialized for seminal fluid production, yet the novelties that we've documented don't seem to serve in that function in the main cells. So it's an interesting paradox, again, a great puzzle that we're trying to hope to get some traction on in the future. Okay, so with that, I want to thank, of course, Julie Cridland and Alex Majane. This work I've talked about is pretty much almost all done by them, and it's really good and careful work. And then I'd like to thank the National Institutes of Health that's funded research in my lab for many years. And with that, I'll stop.